Good day, guys. Good day. I will start that video from the glycogen metabolism. And I know that I said some part of that one in the previous lecture, but the rest of that I would like to make them in one video, to make like one topic because actually this is one topic. So I will talk today about glycogen metabolism. And as we agreed together, what does mean glycogen metabolism? It could be building up glycogen or breaking down glycogen. So we should know, by logic, what time we build up glycogen, what time will break down. So generally, let's start to understand the two process. What does mean building up? What does mean hydrolysis? And how is the mechanism for both of them? Generally, we start with glycogenesis. Glycogenesis or glycogenesis or glycogen synthesis. I mean that we build up. And usually we build up glycogen once we have extra glucose and it should be stored. So we eat, we have too much glucose in our body, we have enough energy. Remember what I'm saying. We have enough energy component like ATP, we have enough glucose, we have too much amount of phosphorylated glucose, then we should store it in the three organs. So the extra glucose can be stored in the muscle or in liver or in adipose tissues. Okay. And we store the glucose in the muscle and the liver in the form of glycogen, while we store the extra glucose in adipose tissues in the form of lipid. The building up process of glycogen happen in two different stages. First of all, you have a glucose, and you remember usually glucose are going to glycolysis. Here is the main pathway. So the first step, glucose, should be phosphorylated like glycolysis by two different enzymes up to where we are talking. If you are converting glucose to glucose 6-phosphate in the liver, there is an enzyme called the glucokinase in liver. If you make this process in the muscle, there is an enzyme called the hexokinase. But both of them adding the glucose phosphate, inorganic phosphate, to the carbon number 6. Inorganic phosphate in the carbon number 6. Perfect. Then that glucose 6-phosphate, guys, that glucose 6-phosphate, by a specific enzyme who can transfer the phosphate group from the carbon number 6 to the carbon number 1. So we have no change in the structure, just to make rearrangement for the phosphate from 6 to 1. From 6 to 1, it is called the mutase, and this mutase can work in opposite direction, from 1 to 6. So this mutase, phosphoglucomutase, which can transfer the phosphate group from carbon number 6 to the carbon number 1. So we have now glucose 1-phosphate. Okay. Where we are either in liver or in muscle. Don't forget, we are in liver or in muscle. That glucose 1-phosphate, now our target is to making one glycogen. And I'm sure you know that glycogen is the highly branched strains of glucose together. Here is our glycogen. Glucose, 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 and they are linking together like branching or straight and so on. So here is our target. But actually, we cannot transfer that glucose 1-phosphate directly in that strains. We have no enzyme who can catch glucose 1-phosphate to add it in glycogen and start to build up. So we need to add something to help to add the glucose to the glycogen. We're using some molecule called, our body use it, called uridine triphosphate. So there is uridine triphosphate coming to bind with glucose 1-phosphate. Then we release 1-phosphate, so staying uridine diphosphate. So uridine triphosphate come to the reaction and bind with the glucose at the carbon number 1 to form UDP glucose. So what is UDP glucose? Uridine diphosphate glucose. Okay. And what is the enzyme who can do this one? It's easy. It is UDP, the UDP glucopyrophosphorylase. So UDP glucopyrophosphorylase, not phosphatase. Phosphorylase, not phosphatase. Because I found some notes on some student notes talking like UDP glucopyrophosphatase. No, it is phosphorylase. So UDP glucopyrophosphorylase added UDP to the glucose to bind with the carbon number 1. Here is the active form of glucose. What does mean active form of glucose? I mean it is the form we can add the glucose in the growing chains of glycogen. But the point is, can I start to add it from here? يعني can I start make a glycogen strain from empty end? No. We need to find something called the primer. يعني we need a primer. Then up to that primer we add a glucose. We need a primer and here we add a glucose. Okay, perfect. So this primer is what? This primer usually I say it is also kind of glycogen. So we need kind of enzyme which able to add a phosphate to itself. يعني we need kind of protein which able to collect that UDP glucose to itself. So that protein is called glycogen. So glycogen, it is glycogen primer. Glycogen is glycogen primer. Remember this one. Glycogen is glycogen primer. That glycogen primer or glycogen able to collect like 7 or 8 UDP glucose. But remember that once you add UDP glucose plus UDP glucose, UDP are released. So you only have glucose. So here is the primer. So this black circle, it is the primer. This black circle, it is the primer. What is the primer? It's kind of enzyme. We call for that enzyme autoglycosylating enzyme. Autoglycosylating enzyme, what does mean? It is the enzyme which is responsible to add a phosphate to itself. Autoglycosylation enzyme using the UDP glucose to glycosylate one of its tyrosine residues. يعني that enzyme has tyrosine and we add a glucose in that one. Okay, so it is an enzyme to make short chain of glucose, like 8. So now we have here this kind of enzyme. Then we have many of UDP glucose. UDP glucose coming to add it here. So here, and each time we add UDP glucose, we eject out UDP. Who can add this one? Who can add UDP glucose to the growing chain? One enzyme called glycogen synthase. So we have one enzyme called glycogen synthase. That glycogen synthase is the enzyme responsible to make straight growing only in one direction. Straight growing like this one. يعني the enzyme who come to add, this is the first one, we add second one, third one, fourth one, and I'm growing in that direction, in that direction. So that enzyme make a link between two glucose in a straight chain. That two glucose are linked together from the carbon number 1 and the carbon number 4. So the link between them is called alpha 1,4 glycosidic bond. So here is alpha 1,4 glycosidic bond between the carbon number 1 and the carbon number 4. Carbon number 1 and the carbon number 4. I hope I'm clear, guys, for this part. So after this, guys, after this one, we can say now that usually here is we are growing from that direction. So we're growing from the 4 and even we break down from the direction. So we are growing from 4, carbon number 4. We're growing from carbon number 4 and we break down from carbon number 4 to back. So we can say that we are growing from non-reducing end. And we are breaking down glycogen also from non-reducing end. That is the carbon number 4. While the carbon number 1, it is reducing end. So we have many of non-reducing end. Here is the function why the glycogen should be branching. Branching means we have many ends which carbon number 4. What does mean? Many ends which can I build up or break down. Yani from that end I can build up and I can break down. So I have many point where I can start to build up or I can start to break down. Here is the function of branching. So you would like to say that glycogen synthase is added the glucose in that direction? Yes. But who can make a branching? Here is the other enzyme. So we need the other enzyme to come this piece. And don't forget, who make link here between this glucose? Glycogen synthase. And who make link between this glucose? Glycogen synthase. But who makes that link? Not the glycogen synthase. Yani the branching link The bond between the carbon number 1 and the carbon number 6 not happen by glycogen synthase. It's the third enzyme. It's called the branching enzyme. So branching enzyme, bring like 7 glucose and add it to here. Branching, 7 glucose. And so on. Am I clear, guys? A branching enzyme go for 7 glucose, bring them to here, and who continue this one? Glycogen synthase. What kind of bond here? It's 1,6,1,6 glycosidic. The enzyme is called alpha-1,4 alpha-1,6 transglycosidase. Remember the name. Alpha-1,4 alpha-1,6 transglycosidase. I hope I'm clear for this part. Okay, so once you're able to make a primer, then you add mini glucose to that primer to make a straight line of glycogen by glycogen synthase. Then you have other enzyme which come to add a branching for that growing one. It is by branching enzyme. Now we have glycogen. And remember, guys, if I'm asking you what's the function and why glycogen are branching, why glycogen is branching? Look, guys, we have for glycogen only one reducing end here. Only one reducing end. But we have many of non-reducing end. That many of non-reducing end, it is the area where we can do glycogen synthase and degradation. So we need many of them for rapid synthesis or we need more mobilization of glucose. So we have many ends. This many ends will help me to make the metabolism is faster. Here is we build up glycogen. Now we build up glycogen. You know what is the primer? It's glycogen, and it is self-glycosylation. It is a protein which can add a glucose for itself. We call it auto-glycosylation. This can make a short alpha-1,4. Remember, alpha-1,4. It makes the same bond like glycogen synthase. What is the bond of glycogen synthase? Alpha-1,4. What is the bond? What can glycogen can do it? Alpha-1,4. But alpha-1,4 can make a short chain, but glycogen synthase can make very long chain. Okay, now we build up glycogen and we know how. So if asking you if you have more glucose, where in muscle, liver, adipose tissues, how it's happened by this kind of two stages. One stage to form active form of glucose, UDP glucose, and second form add glucose to the primer, make long chain by glycogen synthase and the branching in that chain by branching enzyme. Then we talk about the glycogenolysis. Glycogenolysis, it means that we break down where in liver and the muscle. Again, where in liver and the muscle, where we stored. So in liver and the muscle, when we have low glucose in our body, especially during the fasting and the exercise. So you have here one molecule of glucose. You have in one chain link is 1,4 alpha acidic, and the other chain when the branching it's between number 1 and number 6. So here alpha-1,6 is glycosidic and we have alpha-1,4 glycosidic. The one who build the alpha-1,4 glycosidic is called glycogen synthase, and the one who can break down this alpha-1,4 is called glycogen phosphorylase. The one who build this alpha-1,6 glycosidic is called the branching enzyme. The one who will break down this one is called debranching enzyme. I hope I'm clear now. Okay, so the question is to break down this bond. You need to activate glycogen phosphorylase. Yes. So that glycogen phosphorylase, from the name, it seems that it add a phosphate for this one. Yes. So glycogen phosphorylase will bring inorganic phosphate to add it to the carbon number 1. So after I cut here, the released glucose is not free. It is glucose 1-phosphate. Remember, glycogen phosphorylase will cut from non-reducing end. I'm saying what I mean. It's cut from non-reducing end. And the released glucose are not free glucose. It is glucose 1-phosphate. And also also what I would like to explain to you, if we have here kind of glycogen in front of you here, that front of glycogen, guys, we have what? Here is carbon number 4, here is carbon number 1. So what happened here? Usually we come to remove. Who can do this one? Who can do this one? Who can do this one? Glycogen phosphorylase, and each glucose are released here. Is free glucose? No, it is glucose what? Glucose 1-phosphate. Okay, but what I would like to say, I still able to remove, till I have here 1, 2, 3, 4. يعني glycogen phosphorylase is able to remove all the glucose in glycogen except the last 4 glucose. Am I clear for that point? I hope this. Then this last 4 glucose, let me explain it here for you. I know that I give this in the class, but I just would like to make any kind of reminder for you, and we'll see. So here, guys, here is one glycogen molecule, and we have the enzyme which is responsible to remove from non-reducing end, it's called glycogen phosphorylase. It can remove this one, and this one, till we have 1, 2, 3, 4. Remove here, 1, 2, 3, 4. Remove here, and so on, guys. Okay, and so on. So the question now, we are able to release glucose, which is glucose 1-phosphate. Don't forget, glucose 1-phosphate. Who can do this? Glycogen phosphorylase. Then we need other enzyme to come to help us. To help us for what? To remove this, to handle this stuff. So we need one enzyme to come to remove that 3 and add it here. Now we don't have this. So we need one enzyme to come to get this 3 and add it here. I hope you understand. So we got this 1, 2, 3, number 4, 3, 2. We got this one, add it here. So that enzyme which can remove this 3-phosphate, which stayed after the working of the glycogen phosphorylase, and they make a link them again with one strand here. What happened now? Now we have 1, 2, 3, 4, 5, 6, 7. So glycogen phosphorylase will come back to cut from here. So that enzyme is called debranching enzyme. Come to make what? To remove that 3 and add that 3 here. This is called what? Transferase activity. It has a transferase activity. To make what? Oligo, like 3, alpha 1, 4, glycogen transferase. Glycogen transferase. So alpha 1, 4, glycogen transferase. What happened? I got just glucose, 3 glucose from here to add the 3 glucose here. Transferase activity. Okay, so this is one to make, this one is called alpha limited dextrin. In that form, I mean alpha limit dextrin. Here is alpha limit dextrin. Okay, so now here is that one. So what happened after we remove that 3? We remove that 3, we have only one here. That one which stays here, what happened now? The one which stay here should be removed. Who can remove? Debranching enzyme. And debranching enzyme has 2 activity now. Has transferase activity and has hydrolase activity. Transferase to get the 3 adding to the other strand, then glycogen phosphorylase work and remove the last carbon, last glucose. The link here is what? Is alpha 6, is branching point. And the last glucose in that branch will remove as a free glucose, not glucose 1-phosphate. So the last glucose will released as a free glucose. Last glucose are released as a free glucose, not glucose 6-phosphate. Don't forget this one. So all the glucose released by glycogen phosphorylase, are released in the form of are released in the form of glucose 1-phosphate, while the last branching one, last branching one, were released by debranching enzyme in the form of free glucose. Okay, now let's say that generally we have many of glucose 1-phosphate, many. So we removed the glucose from glycogen in glucose 1-phosphate. Let's say that we have one glycogen molecule from 10,000 glucose. So we have around 10,000 glucose in the form of glucose 1-phosphate. Then here you should remember where we are. We are either in liver or in muscle. If you are in liver and muscle, both of them, then you need to convert glucose 1-phosphate to glucose 6-phosphate. Because the glucose phosphate is the one like central one which can I handle it. So who is the enzyme can convert to glucose 1-phosphate to glucose 6-phosphate? Again, it is mutase. So phosphoglucomutase will transfer the phosphate from carbon number 1 to carbon number 6. Now you are in the liver and the muscle and you have glucose 6-phosphate. If you are in the liver and in the smooth endoplasmic reticulum of the liver cell, there is an enzyme called glucose 6-phosphatase. You remember gluconeogenesis? Gluconeogenesis, the last step, when I convert to glucose 6-phosphate to glucose, I need the enzyme called glucose 6-phosphatase. But this enzyme found only in the liver. So only in the liver we can convert the glucose 6-phosphate to glucose. Then the liver cell will send that glucose to the brain. Liver cell will send that glucose to our brain. So while in the muscle we don't have that enzyme not there in that muscle. So the muscle will not convert the glucose 6-phosphate to glucose, but it will go for fructose 6-phosphate and the continuous glycolysis. Sorry guys. So in muscle, this enzyme not there, not in that muscle. So what happened now? So I go to continuous glycolysis to produce ATP, because usually the target of muscle to make glycogen breakdown is to getting ATP. I need energy. I need energy to make my muscle contract. So here we finish this one. So even this slide, glycogen breakdown serves different purpose in liver and muscle. Yes, in liver, my target is to producing glucose to send to my brain, because our brain is only able to feed in glucose, while in muscle we don't have the facilities to convert to glucose 6-phosphate to glucose. So what happened now? In muscle, in skeletal muscle, glucose 6-phosphate will directly continue glycolysis and producing energy from that one. So I hope it's clear till here. So now here is the most important slide for glycogen metabolism. Regulation of glycogen synthase and glycogen breakdown. Look, I'm writing here will not happen in the same time. Yani it's not logic that you build up glycogen and break down glycogen at the same time. So usually when we talk about regulation in physio or in biochemistry, I should ask myself, is that enzyme which working to build up or break down working up to what? Up to my body need. And let's say if you eat good meal, contains carb and protein, fats, too much sugar, for example, drink one cola. You think now your body should go where? Should go to use the glucose and producing too much ATP and the glucose stay in the blood. Not logic. Because I should keep my blood glucose level is constant. The opposite when you are fasting, when you are doing exercise, your body will need to feed itself. I need glucose to my brain to thinking while I'm studying all the night. I need too much glucose to give concentration and focus in my studying, for example. I need glucose to my muscle because my muscle now will working to producing ATP and I need to make ATP for contraction of the muscle. So in that time when I'm exercising or when I'm focusing, when I'm nervous, even when you are nervous, when you are stressful, When you are stressful, you need a lot of glucose. A lot of glucose. So let's say now that we have two main categories or two main enzymes. One enzyme which build up glycogen and one enzyme which break down glycogen. Enzyme build up glycogen and break down glycogen. The one which build up glycogen synthase and the one which break down glycogen phosphorylase. And يعني I make something funny. The enzyme which go to add a phosphate for glucose, it should be phosphorylated. يعني glycogen phosphorylase to be active, it must be phosphorylated. يعني I'm coming to give you some phosphate, it means that I have some phosphate. While the enzyme which working to build up glycogen must be dephosphorylated. So here is the concept. This kind of thing is the concept. To build up, I need glycogen synthase, so I need it dephosphorylated. To break down, I need glycogen phosphorylate, so I need it phosphorylated. Okay, so the step in my body is regulated by hormones. We have mainly three hormones. Glucagon and norepinephrine, epinephrine, and insulin. So mainly we are talking about three hormones. When we have low glucose in our body, so we need to release the glucose. So glucagon will be released. The glucagon released from pancreas. The glucagon go for a specific receptor in our liver cell and the muscle cell. That receptor of glucagon is GS, G stimulatory. G stimulatory will activate AC, increase cyclic MB, cyclic MB activate BKA. BKA protein kinase activation will activate two enzymes. Activate glycogen phosphorylase kinase and glycogen synthase kinase. يعني BKA will active two kinases. One kinase to phosphorylate glycogen phosphorylase and one kinase to phosphorylate glycogen synthase. I think you can continue now. If I add a phosphate to that one, it's working to break down. If I add a phosphate, this one is not working to stop. So I make a phosphorylation glucagon response, phosphorylation for glycogen phosphorylase, and the glucagon will make a phosphorylation for the glycogen synthase. If this one phosphorylated is work, if this one is phosphorylated is not working. So here is the glucagon. What time we release glucagon? When we have less glucose in the body. What time we have less glucose in the body? When we are fasting or exercising. What time, which area in our body affected by glucagon? Liver and the muscle. What is the response of glucagon to make a phosphorylation for the both enzyme? What is the both enzyme you mean? Glucagon phosphorylase and the glucagon synthase. What happens if we phosphorylate both of them? The phosphorylase, if phosphorylated is work. The synthase, if phosphorylated is not working. So what is the result now? I release glucose from the glycogen. Norepinephrine and epinephrine is the same. The receptor of norepinephrine and the epinephrine in the muscle and into the liver, it's also working by beta 2. Beta 2 is GS. Again, increasing cyclic MB, increasing BKA. If I activate BKA, BKA itself will go to activate both kinases, glycogen phosphorylase kinase and glycogen synthase kinase. So now I activate both kinases and you can continue. If I make a phosphorylation for glycogen phosphorylase, I activate it. If I make a phosphorylation for glycogen synthase, I inhibit it. I'm happy because everything is easy. But also discover that there is a receptor for epinephrine. That receptor is alpha 1. So we have alpha 1 which is working as a GQ. GQ pathway, you remember? We remind each other. GQ means we have a receptor signal coming to the G protein coupled receptor, activate BLC, activate BIB2, cut BIB2 into DAG and the IB3. That IB3 release calcium from smooth. So I would like to say that epinephrine increase and norepinephrine result increase calcium. You know what is the effect of calcium? Even from muscle contraction. The calcium which release from the muscle contraction. that calcium itself can activate the phosphorylase kinase. يعني that calcium can activate the phosphorylase kinase even if there is no phosphorylation for it. So they activate phosphorylase kinase without phosphorylation. So what happen? What happen if you activate phosphorylase kinase? Simply phosphorylase kinase will active the glycogen phosphorylase. Clear? Clear. So that two hormones usually glucagon is working when we have fasting exercise, and norepinephrine also with the exercise and the stressful life. So when you are going to the exam and so on, your adrenal gland releases epinephrine, and this epinephrine will go to what? To activate BKA from one side, and the other side, this alpha 1 also release calcium plus the calcium from the muscle. Both of them activate the phosphorylase kinase without phosphorylation, which will activate glycogen phosphorylase. We come for the insulin. The insulin is the guy which working when we have high blood sugar. Eat one chocolate, I like it. I drank like 1 cup of coffee with 2-3 spoons of sugar. What happened for glucose in your blood? Directly increase. But this is not acceptable in our body. Our body should be very fast to decrease the glucose as fast as we can, because increasing glucose above the normal is so dangerous. And this is also related to physio, but second semester for the student in the medicine, second semester you study endocrine, you need this one. And by the way, at the second semester you will need to study this one as well to study this one as well to make the final exam. Anyway, the insulin will generally will activate this guy and inhibit this guy. Activate glycogen synthase, inhibit glycogen phosphorylase. We happy to hear this one. Let me close. I'm sorry. So insulin can do this by 3 ways. By three level of regulation. First of all, glycogen insulin will released. Receptor of insulin is called receptor tyrosine kinase, which will activate many kinases. One of that enzyme which activate after insulin is coming is called cyclic AMP phosphodiesterase. So we have phosphodiesterase enzyme which activated. What happened? Phosphodiesterase will convert cyclic AMP to AMP. So we can say insulin decrease cyclic AMP. And the logic, if I decrease cyclic AMP, I not active BKA, so I'm not activating this 2 kinase. So what happens if you are not activating this 2 kinase? You will make here dephosphorylation, and you will make here, yes guys, I'm not activating the kinases, so I'm not adding phosphate here. What happened for this guy? It's active. So if I clear? I'm not activating the 2 kinases. If I'm not activating the glycogen synthase kinases, I don't add a phosphate here, this one is active. If I'm not activating glycogen phosphorylase kinase, I don't add a phosphate here, is this not active? Am I clear? This is the first level of regulation. Second level, we can say here is number 1. Number 2. What is the enzyme responsible to it will inhibit one enzyme called glycogen synthase kinase. This one. Insulin will inhibit glycogen synthase kinase by 2 mechanisms. One mechanism by decreasing cyclic AMP and the BKA, then I'm not activating this one. Nice. And one mechanism directly I will inhibit that one, because when you send the insulin to the receptor, this will activate something called the insulin receptor substrate, which activate protein kinase B. So this glycogen synthase activated by protein kinase A and inhibited by protein kinase B. So that protein kinase B will make a phosphorylation for glycogen synthase kinase number 3 and inhibit it. Yes. And if I inhibit it, then glycogen synthase not phosphorylate, then it is active. So I active this one as well. Clear, guys? I hope it's clear. Then we'll discuss about the third level of regulation, third level of insulin. we discovered that the insulin come to activate a specific protein called the protein phosphatase number 1. So insulin activate protein synthetase number 1. You know what is protein phosphatase number 1 do? It is remove phosphate. So imagine with me that protein phosphatase number 1 removes the phosphate from glycogen synthetase. It became active. Easy. Okay, and imagine that protein phosphatase go to release the phosphate from phosphorylase kinase. It became inactive. Yeah, if remove the phosphate from phosphorylase kinase, يعني that glycogen phosphorylase kinase is not active. And what's the function of this one? Glycogen phosphorylase function is to add a phosphate to phosphorylate, to add a phosphate to glycogen phosphorylase. So I inhibit the phosphorylase kinase by removing phosphate, and I inhibit directly glycogen phosphorylase by removing phosphate. Here is the step. So I activate glycogen synthetase, and I inhibit the glycogen phosphorylase by removing the phosphate from it and by inactive the enzyme which is responsible to phosphorylate it. Here is the steps and level of, يعني I could say it will be يعني very hard if you didn't understand this here. Last thing about glycogen metabolism, allosteric regulation of glucose 6-phosphate and the AMP. Allosteric regulation of glucose 6-phosphate and the AMP. Glycogenesis increase if I have more glucose, more ATP. Glycogenesis, I build up. If I have more glucose, more ATP, more glucose 6-phosphate. And glycogenolysis increase if I have the opposite. So glucose 6-phosphate itself will result in what? Will result in increased glycogen synthetase, and the phosphatase number 1 is activated in the muscle. Remember, glycogen phosphatase synthetase and activate the phosphatase number 1. So you can expect, so I increase glycogenesis and decrease glycogenolysis. AMP, adenosine monophosphate. If you have more AMP, it means that you have less energy. So AMP itself without phosphorylation can activate the muscle glycogen phosphorylate. يعني AMP can activate this one without phosphorylation, because you know I told you glycogen phosphorylate is active when it phosphorylate, but I can activate without phosphorylation. Who can do this one? AMP and the calcium. We study calcium here. So the calcium and the AMP can activate this one without phosphorylation. And at the same time, if you study glycolysis, I took to you that phosphofractokinase number 1, this enzyme, one of the common steps in the glycolysis. This one is activated by AMP. So look for harmony. Look for harmony in our body. This AMP will release glucose from glycogen, and this AMP will activate the using of that glucose in glycolysis. So I never have more glucose in the blood. يعني don't think that we release the glucose in the blood and then if we measure the glucose level it will be high. No, it won't be high. But what happened, guys? You remove that glucose from glycogen, then you will use it by producing energy from it, by activating pKa in glycolysis. If I'm asking you about the glucose, what happens if we have glucose? Glucose itself will inhibit glycogen phosphorylase in the liver. Glucose will inhibit glycogen phosphorylase in the liver because I have glucose. What about calcium? Calcium released during the muscle contraction. So what happens now? If you have muscle contraction, you need to have more energy, and the more energy means I need to get more glucose from glycogen. So the calcium itself can activate the phosphorylase kinase. Activate phosphorylase kinase without phosphorylation, and activate the glycogen phosphorylase. Okay, guys? So this is kind of response to the physical exercise. So the calcium, also in liver, calcium is released. يعني this calcium in liver by epinephrine with alpha 1. And the calcium in muscle because of the contraction. يعني calcium in muscle and the liver will increase. But the mechanism to increase the calcium in liver by epinephrine with the calcium. The mechanism of releasing calcium in the muscle by contraction. The calcium released in the liver through the fight or flight stimulus. Am I clear? Okay, I hope I'm clear, guys. I hope I'm clear. Yani, let me write in something here about the calcium. Yani, we could say here that, let me do it here because this will help you a lot, I know. Mainly I talk about calcium. You would like to say that the calcium, it increases in where? In muscle and in liver. It's increased in muscle by contraction. You know how is the calcium released in muscle? We study this in physiology in details. In liver, by epinephrine bind to alpha 1, which is GQ. So what happened now? Increasing of IB3, IB3 increase calcium. So calcium will bind with what? Will bind with calmodulin. Calcium will bind with calmodulin. So the calcium will bind with calmodulin. This can go to activate this calcium and the calmodulin. Activate phosphorylate kinase. Hepatic liver phosphorylase kinase. This one activate glycogen phosphorylase. And here is the regulation, what I would like to tell you. I hope it's okay, guys. I hope you understand this part. I will make also the coming videos to you about the pentose phosphate pathway, and I will make in separate video.